Coxsackievirus is a virus that belongs to a family of non-enveloped, linear, positive-sense ssRNA viruses, Picornaviridae, and the genus Enterovirus, which also includes poliovirus and echovirus. Enteroviruses are among the most common and important human pathogens, and ordinarily its members are transmitted by the fecal-oral route. Coxsackieviruses share many characteristics with poliovirus. With control of poliovirus infections in much of the world, more attention has been focused on understanding the non-polio enteroviruses such as Coxsackievirus. Coxsackieviruses are among the leading causes of aseptic meningitis. Groups: Coxsackieviruses are divided into Group A and Group B viruses based on early observations of their pathogenicity in mice. Group A Coxsackieviruses were noted to cause a flaccid paralysis, while Group B Coxsackieviruses were noted to cause a spastic paralysis. At least 23 serotypes of Group A and 6 serotypes of Group B are recognized. Group A. In general, Group A Coxsackieviruses tend to infect the skin and mucous membranes, causing herpangina, acute hemorrhagic conjunctivitis, and hand, foot, and mouth disease. Both Group A and Group B Coxsackieviruses can cause non-specific febrile illnesses, rashes, upper respiratory tract disease, and aseptic meningitis. Group B. Group B Coxsackieviruses tend to infect the heart, pleura, pancreas, and liver, causing pleurodynia, myocarditis, pericarditis, and hepatitis. Coxsackie B infection of the heart can lead to pericardial effusion. Muffled heart sounds and pulsus paradoxus are signs of this. The development of insulin-dependent diabetes has recently been associated with recent enteroviral infection, particularly Coxsackievirus B pancreatitis. This relationship is currently being studied further. Sjogren's syndrome is also being studied in connection with Coxsackievirus, as of January 2010. History: the Coxsackieviruses were discovered in 1948–49 by Dr. Gilbert Daldorf, a scientist working at the New York State Department of Health in Albany, New York. Daldorf, in collaboration with Grace Sickles, had been searching for a cure for poliomyelitis. Earlier work Daldorf had done in monkeys suggested that fluid collected from a non-polio virus preparation could protect against the crippling effects of polio. Using newborn mice as a vehicle, Daldorf attempted to isolate such protective viruses from the feces of polio patients. In carrying out these experiments, he discovered viruses that often mimic mild or non-paralytic polio. The virus family he discovered was eventually given the name Coxsackie, from Coxsackie, New York, a small town on the Hudson River where Daldorf had obtained the first fecal specimens. Daldorf also collaborated with Gifford on many early papers. The Coxsackieviruses subsequently were found to cause a variety of infections, including epidemic pleurodynia, and were subdivided into Groups A and B based on their pathology in newborn mice. The use of suckling mice was not Daldorf's idea, but was brought to his attention in a paper written by Danish scientists Skov and Andersen in 1947, who were using such mice to study a mouse virus. The discovery of the Coxsackieviruses stimulated many virologists to use this system, and ultimately resulted in the isolation of a large number of so-called enteric viruses from the gastrointestinal tract that were unrelated to poliovirus, and some of which were oncogenic. The discovery of the Coxsackieviruses yielded further evidence that viruses can sometimes interfere with each other's growth and replication within a host animal. Other researchers found this interference can be mediated by a substance produced by the host animal, a protein now known as interferon. Interferon has since become prominent in the treatment of a variety of cancers and infectious diseases. In 2007, an outbreak of Coxsackievirus occurred in eastern China. It has been reported that 22 children died. More than 800 people were affected, with 200 children hospitalized. Cavitac, a wild-type Coxsackievirus A21, is being used in human clinical trials as an oncolytic virus. References. External links: 3D macromolecular structures of Coxsackieviruses from the EM databank, EMDB.